Hello everyone, I'm excited to have the opportunity to introduce you to the Virus Pathogen Resource and tell you about the data and the tools that we have available for studying vector-borne viruses. You can find us by going to viperbrc.org. And like VUPathDB, we're funded by the NIH's NIAID department, and we belong to one of the bioinformatics resource centers. And we focus mainly on the NIAID's list of emerging and re-emerging pathogens, which can be found on the BRC page if you're interested in exactly what we look at. And these cover mainly category A through C pathogens. Our partners at the Epitope Database focus on bacterial pathogens, whereas we focus on viruses, including a website dedicated specifically to studying influenza virus, the Influenza Research Database at fludb.org, and our Virus Pathogen Resource Database, which looks at multiple other virus families, which are summarized in this section of the homepage, including single-stranded RNA viruses, both positive and negative sense RNA, double-stranded RNA, and double-stranded DNA viruses. And families of particular interest to this audience would of course be the Flaviviridae and Togaviridae families, as well as possibly some of you might be interested in Bunia viralis or Rheoviridae. If you scroll down a little bit further, you can see that we also offer specific pages and tools that focus only on viruses that have been the subject of recent outbreaks, and these of course include the two Flaviviridae, dengue virus and Zika virus. If you're looking for a specific group of viruses, you can also scroll down and search through our taxonomic tree. Let's say, for example, you're interested in dengue, you can open Flaviviridae, click on the flavivirus genus, and search through all of the different species and strains that we have available. And what's nice is that the ViPR database lists exactly how many strains are available, and out of these, how many of them are considered to be complete genomes. So going back up to the top of the page, you can basically think of the website architecture as having three different sections. The first is the search function, where you can search and explore and browse the different types of data, including sequences, strains, immune epitopes, 3D protein structures, and multiple other things that might be available for the specific virus family that you're interested in. The second is the analysis section, where you can explore the different tools and bioinformatic pathways that are available for different viruses. And we also have a virtual workbench where if you're a registered user, you can store your saved data sets, you can store any analysis that you perform on this website, and you can also upload your own data and integrate it with the ViPR data to again perform analysis on these combined data sets. If you're interested in what types of data we have available for each virus family, you can go to the About Us section and click on the summary of ViPR data. And this will take you to a page where you can see the different types of information that we aggregate based on each viral family. And I encourage you to browse through this section on your own. So going back to the webpage, if you're interested in where we get our data, you can again go under the About Us section and click on the sources of ViPR data to see the different databases from which we aggregate our own data. And not only that, we also generate our own data, not just collected from other resources, including mature peptides, standardized annotation of viruses, calculate isoelectric points, and sequence feature variant types. So going back to our webpage, let's say you're interested in browsing the different types of data that are available for, let's go ahead and click on dengue virus. You can then either click on search data and click on the specific data that you're interested in looking at, or if you just want to browse everything that we have, you can click on browse all search types. And this takes you to a page and shows you the multiple different things that we have available. We can search for sequences and strains, both nucleic acid and protein sequences. If you're interested in immunology of the virus, you can look at immune epitopes. We have a list of antiviral drugs that's available. We have some host factor data, but for now these are limited to influenza and coronavirus. We're hoping to expand them to other virus families as well. If you're interested in the structural biology of your virus, you can look at 3D protein structures and protein domains. We have a little bit of plasmid data, and you can also search for sequence feature variant types. So for example, if you're interested in looking for a specific structure in a protein or a specific feature in a protein. And finally, we also have one of my favorite things about the dengue virus page is our human surveillance data, which will show you viruses that have associated with them human surveillance records, which show you detailed host clinical metadata. If you go ahead and click on this, you can see that there's quite a lot of detail associated with these sequences, including information on the virus, the hosts, as well as clinical outcomes and attributes. If you're interested in the tools that we have available, you can click on Analyze and Visualize, which will show you a list of tools that we have available for traditional bioinformatic functions such as sequence similarity searches, PCR primer design, or sequence format conversion. We also have annotation tools, as well as comparative genomic analysis tools. For example, we use Muscle for performing multiple sequence alignments, and we have multiple sequence alignment viewer, which helps you visualize your alignment and modify it online rather than having to download it into your own software and manipulate it there. We also have two different tools that look for sequence variation. One of them is a traditional SNP tool that looks for sequence nucleotide polymorphisms, and it can look at variation both in amino acids and nucleotide sequences. And we also have the metadata-driven comparative analysis tool, or MetaCats for short, which allows users to perform statistical analysis on positions that might differ based on two groups of viruses that have different metadata attributes. So for example, one group might be from the current year, whereas the second group of viruses might be from the previous year, or one might be from Asia and the other might be from Africa. And this tells us whether there are any positions that are different and have a statistically significant difference between the two groups. So I find that very handy. And of course, we also have tree building tools, including neighbor joining and maximum likelihood methods. We also offer the model compare tool, which helps you choose the best evolutionary model to build your tree. And we also have a really nice interactive tree viewer, which helps you annotate your trees and different branches based on the different metadata that's associated with those viruses. So you don't have to do that manually. This software will do it for you. And we also have a tool that helps you characterize the genotype and subtype of your virus. So moving on to describing what your workbench does for you, if you go ahead and click on Workbench, you do have to sign up for this with your email address, and I would encourage you to do that because you can then save your data in your workbench and run further analysis on it. You can also upload your own data to your workbench and use all of our bioinformatic tools. You can combine your own data sets with anything that's available in ViPR and run your analysis on these combined data sets. And what's really nice about it is that you can also share this data or the results of your analysis online with any of your colleagues or your lab mates. You can create shared folders. This is really helpful for those of you who have international collaborators and you don't want to just spend time transferring large data sets. And it's extremely handy in countries where low bandwidth is a problem. So that's it for my quick introduction. I hope you found this useful, and thank you for your attention.